Here we go. All right. Thank you, Delia. As Delia said, I'm Nicole Goche. I am the plant pathologist for specialty crops at UK. And by specialty crops, I do fruits and vegetables. We just heard, a little bit about strawberries in a high tunnel. I am the pathologist for fruits as well. But today we're talking about disease. In fact, we're talking about kind of disease 101. So we're going to go right down to the basics. I know diseases are a little bit harder to understand because pathogens, of course, are microscopic. And with a microscopic pathogen means you just really can't see the causal agent. Disease, as we use the term, it's the symptom. So when we talk about plant disease, that's a symptom that the plant is showing, not the actual pathogen causing disease. That can be a little bit tricky. Sometimes people don't really realize. So when we talk about leaf spots, that is a symptom, that's a disease. And for example, lots of different bacteria and fungi can cause leaf spots that may all look the same. But we're going right down to plant pathology 101 today. And for those of you who do the in-person, we'll in the next couple of months, we'll really drill down a little bit more detail. But anyway, so disease 101. Well, anybody who ever studies plant disease knows what we call the disease triangle. And this is a very old concept. There's no better way to describe than to still use that disease triangle example. The premise here is that in order to have a triangle, you must have three sides. And if you break one side of the triangle, you no longer have a triangle. So if we talk about disease in that very same way, if disease were a triangle, we have to have three components or we no longer can have disease. Okay, so three things must always be present. We must have the pathogen. That's that microscopic organism that causes disease. So those are fungi, bacteria, viruses, nematodes. We could go on with some parsing it out, but fungi comprise about 85% of our disease agents. Bacteria would be number two, and then viruses. So those are the ones you would see the most. So without a pathogen there, you don't have disease, right? So you can't have Cercospora leaf spot if Cercospora, the fungus Cercospora is not present. So as I kind of go through this, think about management as well. And I know as growers, you're thinking management. So if we can get rid of the pathogen or prevent the pathogen from being there at all, that would be an approach to management. So we all think about fungicides, but what about pruning? What about tilling and burying your inoculum? What about sanitation? So those are all ways that we would eliminate or prevent the pathogen from being introduced. And I'll talk about each of those like individually. The second side of the disease triangle is the host. The host is, of course, your plant, your crop. You have to have a susceptible host in place. And sometimes it's a genus or species that is susceptible to a certain pathogen or infection by a pathogen. Sometimes it's a plant part. Some pathogens don't infect until, for example, the reproductive stages, or some will only infect young leaves and not older leaves. So the susceptible plant or plant part has got to be in place. So we have to have the pathogen, a susceptible host, and the third side of the disease triangle is a favorable environment. And what we mean there is we have to have moderate temperatures and free water. Moderate temperatures is what you and I call moderate as well. If you're comfortable, the pathogen is comfortable. So not too cold, not too hot, not January and not August. So moderate temperatures really mean usually 65 to 80 degrees Fahrenheit, or usually our ranges where we see the most plant disease optimal conditions. There are some that can tolerate hotter temperatures, some can tolerate colder temperatures, and of course, we've always got microclimates. Microclimates might be a shady part of a field or a greenhouse or a landscape. It can mean the inner canopy where there is shade or maybe moisture soil. So when I'm talking about temperature, I mean in microclimates. Okay, so the second component of that favorable environment is free water. And free water is everything from overhead irrigation to rain to fog to dew to high humidity. And if you remember anything today, I want you to remember the magic number of 70%. That is your threshold for most of our plant pathogens. If you can keep your relative humidity below 70% in your high tunnel, you will deter a lot of plant pathogens. Okay, magic number 70%. There are some that require higher, only one requires lower. So if you remember 70, you're good to go. And we'll talk more about that today, and in particular in our in-person workshops. All right, so let's take a look at each of those sides of the disease triangle separately. Let's talk a little bit more. Let's drill down a little bit more. We're going to start with the host plant. So remember, that is that one side of the disease triangle. So when I say host, I don't just mean the plant itself. We can mean leaves. So some pathogens will only affect leaves. Some will only affect stems, some only trunks. Now we're talking tree fruit, and some will only infect roots. So that susceptible host plant part has got to be there. Sometimes they will only infect new growth. Okay, so emerging new growth. Powdery mildew is one. That's when it's infecting. It might not be when you see it, but that's when it's infecting. So infection is different than that disease development. Some pathogens will target overripe fruit. We see a lot of that. Every pathogen, we're not going into 5,000 pathogens and memorize their conditions and their host susceptibility ranges today. But keep in mind that when we talk, a lot of times you'll see me talk about cultural practices, and we'll also talk a lot about the biology or the life cycle of certain pathogens. So if you get a diagnostic report, we'll usually attach some fact sheet to it, and there will always be some life cycle, and I'll describe how that pathogen overwinters, how that pathogen survives and infects. Those are very important in terms of disease management. So we're not just trying to throw a lot of science at you. It really does matter. Okay, so also within a host, so within some genera can be resistant or susceptible. Sometimes it's down to species level, right? So and then other times it's cultivar. So we've got a lot of breeders who are breeding cultivars for resistance or tolerance. Tolerance in most of the time we mean semi-susceptible. Reading your seed catalogs, for example, you'll usually see some listings of some disease resistance in certain cultivars. ID 36, our vegetable spray guide, which I hope all of you are familiar with that publication. It's not just a spray guide. It's not just full of fungicides and insecticides, but there are lists of cultivars that are recommended for Kentucky. So here is an example. Here's a screenshot right from the tomato. These are field tomatoes. This in particular are the large red ones. So you'll see there which of these cultivars have some resistance for, I'm looking at Fusarium, race 2, Verticillium, wilt. So any kind of susceptibility or resistance in particular will be listed on some of the more common varieties. And again, your seed catalogs and your seed distributors will have that information as well. So if you have a history of a disease in your field or in your high tunnel, you're always going to look for resistance. So that is your starting point. Okay, so also as we talk about hosts, vigor is really important. So healthy plants are much more tolerant to disease than weak or wounded or stressed plants. In particular, think about wounding. So wounding is kind of like an open door to for these pathogens. They can get in when there is a wound or something to let them in. This carrot here, that's black root rot. So usually some nick or wound, whether it's mechanical with a mechanical hoe, sometimes insects, sometimes just just environmental conditions, enough stress will cause cracking or some type of opening that the pathogens can get in. So wounded or stressed plants are much more susceptible to infections than healthy, vigorous plants. So a lot of times you'll hear me talk about things like stress pathogens or opportunistic pathogens. There are a lot of those, especially in the fungal world. So an opportunistic fungus, for example, is one that's just waiting for you to open that door and let it in. So they're not aggressive enough or pathogenic enough to penetrate an external cuticle, for example. But once there's some type of wound or stress event, they're ready. They're ready at the waiting. And I can't say enough about wounding. This is an example of botrytis on onion. So you'll see here, my cursor. You'll see here botrytis coming in, and botrytis is gray mold, by the way. Most people know it by gray mold. So this onion was nicked by a spade, and then botrytis, the fungus, it entered, it was able to get in, and botrytis loves wounded tissue. It loves dead or dying tissue. So it started to colonize first that wounded tissue, and from there it gains a foothold, and then it can actually penetrate that onion. So we see a lot of those types of interactions, these pathosystems, in a lot of specialty crops and a lot of agricultural crops as well. Okay, so pH imbalances, nutrient deficiencies, those are all plant stresses. And plant stresses, again, can cause just weakening, cracking, whatever kind of a low vigor event. This is another botrytis fruit rot on these tomatoes. And this image in particular was from a stunted, weak tomato plant in a high tunnel. But it just in general, think about weak and low vigor plants will be more susceptible, especially to opportunistic pathogens and those with low pathogenicity. Sometimes too, think about over-fertilization, so excessive growth. And so when a plant is growing really fast, it is also maybe less vigorous or at least weaker. You've got a longer period where you've got that new tender tissue that is easier for pathogens to penetrate. This is Phytophthora root rot that has moved up into the crown. And this was, and Phytophthora commonly comes in or is more severe in the type in over-fertilized or plants that are growing excessively quick. We see this a lot in fruit crops as well. For instance, fire blight in apple. It's infecting tissues in that growing tip, in that meristematic tissue. So those are, again, examples of how these pathogens can get in. Okay, side 2 of the disease triangle is environment. That with those favorable environmental conditions that I talked about. And this is the one that we can often control pretty well. So when we talk about environment, we're talking about humidity, soil moisture, fertility sometimes, and then temperature. So it's usually temperature and water are usually the two big ones that we talk about. And with these moderate temperatures and free water, pathogens that are there or are easily able to infect and to complete their life cycle. That means to produce spores. The spores can be disseminated and they can reinfect. So that whole life cycle will occur if you've got that the temperature and the moisture conditions. So we're going to start by selecting a good site. We're going to think about where dew settles. We're going to think about maybe a shaded part of your high tunnel. Maybe there's a leak, maybe again, a low part of the tunnel where you don't have good drainage and the soil stays moist. And then also we're going to start thinking about how we space our plants in our high tunnel. How wide are our rows? How much are we spacing plants within a row? And when we prune and stake and tie, are we letting air circulate? So air circulation is the way we're drying out our leaves, or getting rid of that free water, that free moisture, lowering that humidity by air circulation. So the more we can eliminate some of those microclimates or those very humid conditions, the more we can prevent epidemic levels of plant disease. Think too about microclimates. Microclimates are just inner canopy, lower canopy, anywhere that we would have, it would be cooler, maybe shadier, and also to have more moisture hanging there. This image is southern blight, and southern blight likes it, actually it likes it hot, and it likes moisture. So in this black plastic condition, there's a lot more water, it's drying out more slowly, and of course it's hot under that plastic. So this black plastic created a microclimate for this Athelia rolfsii is the name of the pathogen. And so we tend to see, so if there is a history of southern blight in a high tunnel or in a field, and you put plastic, it just creates a better environment for that pathogen. Now, of course, if you don't have that pathogen in the soil, then a plastic culture system it's a different conversation. But in this particular event, if you had a history of southern blight, well, then that pathogen is happy underneath the plastic. So think about it in those terms. There are lots of different pathogens, lots of different scenarios. I'm just giving you examples. Okay, so drainage. So soil moisture is it relies heavily on drainage. You have to have good drainage, both surface drainage and internal drainage. Raised beds are always helpful. We see a lot of high tunnels where you do have a lower end, or maybe when you tilled, maybe it's not flat. There may be some dips where you can get some heavier soils, or you can get some puddling. So raised beds will help you eliminate a lot of that. Making sure you have good drainage will keep that microclimate less favorable to soil-borne pathogens. So wet soil means that humidity and that free moisture for pathogens. Okay, and then, just things, these are horticultural type things. Having your pH in check, having your soil structure in check, proper irrigation, and those are things that Dr. Rudolph has talked about. But again, any stressful situation is going to be conducive for diseases. So especially your opportunistic pathogens that are waiting for an opportunity to infect. So I'm not going to go into any of that, but again, I said this earlier with the host plant, but healthy plants mean less susceptibility to disease. Okay, and the third side of the disease triangle is the pathogen. Pathogens are the fungi, bacteria, viruses, nematodes that cause disease. So all of our plant pathogens are microscopic. They're very small. And so it's very rare that we actually see a plant pathogen. We see disease. We see the symptom after infection occurs. Let me also, if you're going to remember, point 2 to remember today is infection can occur days, weeks, sometimes months before we see the symptoms. So when we talk about, so we rarely talk about reaching a threshold or scouting before we spray, for example, or before we try to manage disease. Instead, our whole focus when we manage disease is to prevent infections from the start. And that's really critical, is to understand when infection occurs. You see a lot of that in our fact sheets and our information. We try to tell you when either the plant is in a susceptible growth stage, or when the pathogen is reaching its ideal environmental conditions. And that is when you have to start managing, whatever manage means to you. For some crops and some pathosystems, it might be fungicides. Others, it might be time of planting might be different. It might be rotations. It might be pruning, etc. So think about it in those terms. This is anthracnose on beans. So the symptom you see is, are these leaf spots. but see all this little orange inside these leaf spots? Those are actually, those are spore masses. So that's what we would call a sign. So you're looking at the actual spores and the kind of gelatinous ooze that the spores are contained in, but the actual spots, the sunken spots with all the discoloration and the necrosis or the dying tissue, those are all symptoms. That's what we call disease. Okay, so there are two different ways that we can think about how the pathogen fits into the disease triangle. We can start by excluding the pathogen and not letting it into our high tunnel or our field from the start, or after infection occurs, then we're going to try to eliminate it. Okay, so let's talk first about exclusion. Let's keep it from coming in from the start. Well, that starts with clean plants or clean seed, being very careful what comes in from the very start. So just because a friend is sharing seeds that he saved from last year, or just because maybe one of your distributors is cheaper, maybe you think you get a bargain, be very careful with what you bring in. Transplants in particular, but pathogens can be seed-borne as well. You can bring a lot of pathogens in by seed. So clean plants, if there's a certified program, whether certified seed or clean plant program, that's where you're going to start. Secondly, you're going to inspect. You're going to inspect whatever you're bringing in, whatever shipment, whatever purchase you make, making sure that you're not bringing in seeds with spots or discolorations or wrinkles, or plants with unhealthy root systems, or any kind of cankers or spots on the leaves. You're going to examine those very carefully. And if you are bringing in transplants, you're going to pop a few of those out and you're going to look at the roots. So making sure you start on your best foot and not bring in anything that's unhealthy. Okay, so then once the plants are in the ground, think about how you may have some soil-borne pathogens or pathogens that overwintered in debris, for example, on that site. So how do you keep them from moving onto the plant? Preventing splash, those types of things will keep your plant healthy, even though you may have some history of disease. That's a whole separate lecture, but using mulch, whether it's organic mulch or plastic mulch or whatever you're going to use, but to prevent soil from splashing onto plants, will keep any propagules from splashing as well. And then just sanitation, just keeping things clean. Sanitation means in the plant world just what it means to us as humans. We're keeping things clean, and by making sure we don't leave debris around, we're keeping that pathogen. So a lot of pathogens overwinter or over summer in debris. So you're cleaning all of that up. Also, you're flushing all of your irrigation lines. If you're going to use the same weed mat, you're going to wash that down with maybe one of the peroxide products. Okay, so a couple of things that I want to say before moving on is that a lot of plants may be hosts for a disease that you maybe relate to a certain vegetable crop. Okay, so weeds and volunteer plants and other volunteers from a previous crop can serve as a green bridge. They can serve as alternative hosts. So making sure that you have managed weeds. You're not just managing weeds for insects and just to keep things tidy, but weed management also matters in disease management as well. So making sure that volunteer plants and weeds are out of there and just cleaning up any debris. If you have dead or dying plants or dying tissue, you're pruning it and clipping it all along the way, not throwing it in the center of aisles, not leaving it there. And at the end of the season, when you terminate your crop, you get it all out of there as soon as possible and get it away. So your debris is going to harbor a lot of pathogens. That's how a lot of our, especially fungal pathogens, overwinter. So if you keep those clean from the start, when your new crop comes in, you won't have any problems, or you'll have fewer problems with disease. I want to point out this this image here on the right. That is a Rhizoctonia. That's a root infection or a collar rot. So just really quickly, so this is a spot, right? Obviously, it's a spot. I'm calling this one a canker because it's on a lower stem. You'll see the roots developing. But look at the margin between that spot and see kind of you got this discoloration before you get back into the white again. That is pretty typical of when you have a plant disease. So the fungus in this case, this Rhizoctonia, is running ahead. So the center here is the oldest part of the spot. And so you've got the death, and in here, you're going to have a lot of different fungi coming in after the fact. But here on the margins, I've lost my cursor again, sorry. But here on the margins of this canker, this tan discoloration, that is where the fungus is running forward. So the fungus keeps moving, it keeps killing as it goes, and it keeps moving forward. So in this particular case, there are two spots, but at some point they're going to meet in the middle and they're going to coalesce. So as you think about fungal leaf spots or cankers or root lesions, we could just use the word lesion in general. You've got this constantly moving fungus, okay? And they are going to keep moving as long as conditions are optimal. And remember, optimal means free water and moderate temperatures. So what's going to happen when it gets really hot or really cold, then it's going to just slow down that movement. It's not going anywhere. It's not dying. It's going to just slow down its movement. But in this case, for instance, if you get a really hot spell, well, then you've already lost a lot of the stem area. So water can't move. Water and nutrients can't move up to the upper parts of the plant. So that's why you'll see a collapse, or you'll see more symptoms in the hot summer, for example. But some of these infections are starting much earlier. Okay, and here's a close-up actually of those same cankers. So again, you've got a lot more damage here on this kind of this lower stem and root area. Oh, I'm sorry, this is Fusarium. It's not the same one. It's a different one, but very much the same thing. So think about what happens when you start losing root mass and you start having that girdling of stems and lower stems. You're going to have that collapse during the summer. You're not going to always see those symptoms when you have sufficient water and cooler temperatures. So sometimes you've got that delay in that symptom development. So I just kind of wanted to mention that. All right, so once you have disease, once a plant is diseased, first of all, there's no curing it, okay? You cannot cure plant disease. Fungicides do not cure. That's point number 3 of the day. Fungicides don't cure. Once a plant is infected, it is infected. If it's something as simple as a leaf spot, you can suppress, you can prune, you can maybe salvage the plant to the end, to get a little harvest out of it, but you're never going to cure the plant. All right, so once a plant is infected, think about where the infection is and what's happening. If it's something like a canker on a stem or a crown rot or a root rot, there's nothing you can do. So would you consider removing the plant as quickly as possible? Would you consider pruning it out? In terms of fruit, these rotted fruit here, you're going to get those fruit as far out of your high tunnel as possible. You're not going to leave them there. You're not even going to wait them out. You're going to get them out. We talk a lot about something we call clean picking. And so always picking in 2 different buckets or 2 different baskets. One is always going to be the damaged fruit that you're going to throw away. The other one are the clean ones that are going into the cooler or to wash. You're never, ever going to leave anything damaged in the field, because again, that's that overwintering or surviving inoculum in debris and eventually in the soil that will be there for the next year. So here's another image. This is gummy stem blight on watermelon, I think. So you've got this infected stem. You're not going to cure this stem with fungicides. So pruning this out and getting this out of there, maybe other vines on this plant are okay, but this one is not. So pruning that out and getting it out of there. Now, smaller growers are much better able to do things like that, pruning small amounts, as opposed to very large growers just won't be able to parse it out as easily. But any debris, anything you cut or trim or clip or anything that looks bad, cut it out, get it out. I also like to drag a tarp between rows. So as you prune, you can throw things on the tarp, fold the tarp up, and then drag the tarp right out. So keep it clean. I mean, these are all layers of the sanitation principle. Keep it as clean as you can. And that way you won't have that buildup of inoculum. Things won't get worse as fast as if you leave it there. And, speaking of keeping it clean, keeping your tools clean, any containers you use, if you have a history of disease, think about how you could use maybe a foot bath. But if for soil-borne pathogens, those pathogens, those propagules, are mixed into soil particles any way you move soil. Any of your bacterial pathogens in particular are usually in plant sap or any kind of ooze. They will stick to your tools and your pruners, your knives, your hands. So any way that you can move these pathogens around, again, think about sanitation in your house or in a hospital. It's the same thing in your high tunnel. So if you have a history of disease, you're going to ramp up your sanitation program. If you don't have a history of disease, you still need to understand, you still need to be cautious. You're not going to be careless. But if you have a history of disease, keeping things clean is of utmost importance. You in particular don't want to move pathogens from, say, a house with disease to maybe another high tunnel that is clean. So think about how you would move your tools, how you would move your equipment, whether it's wheelbarrows or a tiller. Think about think about your shoes. Are you going to move from early in the morning? Will you work in the clean house first, then the dirty house, and then you'll go inside and shower and wash your boots, wash your equipment? Or if you do have to swap, to make sure you're washing things between tunnels, for example, or between crops, depending on how you have everything set up. For those of you with greenhouses, think about which different crops you have in which areas of the greenhouse. So pathogens move a lot of different ways, not just dissemination by wind, but we also move them around with soil and with sap. Okay, and then finally, roguing. If a plant doesn't look good, if a plant is diseased, get it out as fast as you can, throw it away, cut your losses, replant, or just consider that you are not going to be able to have that plant there, because the more heavily diseased a plant becomes, the more infective it is, and propagules, whether those are spores or bacterial cells or whatever the propagule is, can spread to healthy plants. And if you have a history of disease, rotating. I suggest rotating anyway. I'd like to see you rotate at least on a 3-year basis. But if you have a history of disease, rotating becomes even more important. So we have some suggestions for rotations. Depending on what disease you have a history of, will depend on how many years and what type of alternative crops you can bring in. And that's what we're here to help you with. Your county agent helps you with those things. We help you with those things. So think about rotations always. But if there is a history of disease, make sure that rotations are part of your disease management strategy. Okay, so recap. What all did I talk about? I rambled on about a lot of different things, but in particular, the disease triangle. Your goal to manage disease is to break a side of the disease triangle. You can eliminate the pathogen by roguing, by pruning, by not bringing the pathogen in the first place, by fungicides. You can manage with the host plant by using either a genus or species that is not susceptible, and we would use that in rotations, or you would use resistant cultivars. Use crop timing to have the susceptible tissues coming in at different times than the pathogen. And finally, your your moderate temperatures and free water, humidity below 70%, you are keeping the environment that's not, so it's not conducive for infection and sporulation. All right, so maybe some of you have been a little bit antsy because I haven't talked about fungicides yet. This is not a fungicide talk. So before using fungicides, or even if using fungicides, you still have to consider cultural practices. All of the things I talked about that I wove into the disease triangle topics, those still apply whether you're using fungicides or not. Always begin with resistant cultivars if they're available. Using cultural practices, in particular sanitation, time. If you're going to use fungicides, you have to time them. You have to know what you are spraying for. You're not going to use a symptom to diagnose your own plants. We have a plant disease diagnostic lab, and your county agent can help you with that. You're always going to manage soil moisture and humidity, and if possible, microclimates, so that you will have an environment that is not conducive for disease. Sanitation. Through the season, you're going to prune and clip, and you're going to haul debris out. And after harvest, you're going to clean things up very well. Then you can talk about fungicides, right? I want to add a couple of things about fungicides. First of all, I hope you're all familiar with ID 36. This is our spray guide. This is what it looks like this year. It's a 2-year spray guide. We think this one's going to actually go into 2024, but this is a spray guide that summarizes all of the products that are available. But high tunnels are considered greenhouses when it comes to pesticides. So any fungicide, insecticide that you are going to use for your high tunnel must be labeled for greenhouse use. Okay, that really limits the number of products that are available for you. We have another guide, the Southeast Vegetable Workers, extension workers, sorry. And there is a whole greenhouse, there's a really nice greenhouse section in the back. So your county agent can help you with that one as well. So both guides available. These spray guides are our summaries that we do every year or every 2 years. But guess what? Printing occurs once a year or every 2 years, and labels change very quickly. So ultimately, you are going to refer to your label. Okay, our spray guides are guides. Your label is the law. Okay, so that's it for me. I want to point out our website, plantpathology.ca.uky.edu. That CA means College of Agriculture, by the way. So that's where you're going to go to find a lot of our fact sheets on specific diseases. And then our social media. I know you've seen some social media sites pop up over the last couple of sessions, but Facebook and Twitter. So I have both a disease page, and our Fruit and Vegetable Working Group have a combined social media platform. So Fruit and Vegetable Extension is what we call it, or Kentucky Fruit and Vegetable Extension. And then I do want to point out our newsletter, Kentucky Pest News. Even if you just type into Google Kentucky Pest News, you should find it, and you can subscribe to that. And that is a weekly newsletter that's going to cover a lot of pest management topics for all crop types. So with that, you know what I'm going to do? I'm going to keep sharing my screen here for just a little bit, and I can take questions. Oh, thank you, Rachel, for putting the SevU link in there. We needed that.
And Nicole, thank you for that. You do have one question, and I don't know if you can see it, but he says, in terms of debris causing disease, I used to always compost my old tomato plants, corn stalks, etc. Should I stop doing that, or is composting debris okay?
Okay, that is a really good question. So composting, I'm a composter too, so let me just start there. But in my work capacity, I usually come in when there is a disease problem. So you should not compost disease materials. And Commercial composting facilities can get their compost to 160 or 165 degrees Fahrenheit, which is enough usually to kill a lot of our pathogen propagules. Even if you were a farmer, you're still not going to have a hot enough compost pile to decompose a lot of these pathogen propagules. So if it's diseased, do not compost it. If it is not diseased, I highly favor composting. Let me add just a little bit about these propagules. I keep calling them propagules because sometimes it's just a spore, and other times, like some of our pathogens create these like capsules, melanized capsules that have spores inside. And when propagules are melanized, they become heat tolerant, UV resistant, cold tolerant. So their survivability is much higher. So they're really tough, is my point. A cold winter doesn't kill off disease or pathogens. A hot summer just puts them into a dormant period. So they're pretty tough once they have gone into that dormancy or that latent phase. So don't assume that just because you're composting and breaking down plant tissue that you've also destroyed those propagules. So unless your compost pile gets very hot, and rarely do our compost piles get hot, don't compost pathogens, anything diseased debris.
Great, thank you for that. And for those of you all that are still on here, I am going to launch our last poll. If you all could take just a second and fill that out based on Dr. Gaucher's presentation. There's all kinds of really good information links in the chat box. If you look at that, Southeast Veg Guide, ID 36 that Nicole talked about, and then as well as the link to the Extension Vegetable Crop Research YouTube channel. The recording for this presentation will be on that same site. So definitely bookmark that and look for this webinar either tomorrow or maybe Monday. And then also don't forget that we do have 3 in-person workshops coming up. They are all covering the same topics. So we are going to be lucky enough to have Dr. Rachel Rudolph, Dr. Nicole Gaucher, Dr. Jonathan Larson as our specialists at these workshops. So you will receive an email about registration, how to register for those, and then you'll also receive an email with directions and the address to the place after that as well. All right. So I think we will go ahead and close that poll out. And again, thank you so much to all of our speakers on here, USDA, NRCS, and FSA. And yeah, keep checking back on our YouTube channel for more videos and more information. And thank you all so much. Have a good afternoon.